Hello and welcome again to the microbial world. In this episode, we are going to be studying about selected processes involving microorganisms, alcoholic fermentation, wine, beer and distilled spirits, vaccines, yeasts. Kindly subscribe to this channel, like and comment. Alcoholic fermentation is a biochemical anaerobic process carried out predominantly by yeasts, especially Saccharomyces cerevisiae, and occasionally by some bacteria such as Zymomonas mobilis. During fermentation, sugars are converted into ethanol, carbon dioxide, metabolic energy ATP, the overall simplified reaction, glucose ethanol plus carbon dioxide plus energy ATP. Biology of yeast fermentation. Glucose metabolism occurs into key stages. 1. Glycolysis Embden-Meyerhof-Parnas pathway. Glucose pyruvate occurs in the cytoplasm, produces 2 ATP and 2 NAD. 2. Alcoholic fermentation stage. Pyruvate acetaldehyde ethanol. NADH is oxidized back to NAD, allowing glycolysis to continue in the absence of oxygen. Wine fermentation. Raw material. Grapes are the primary raw material. Their sugar content, acidity, and tannins influence flavor and alcohol yield. Sugar concentration, typically 180 to 240 grams per yeast used, Saccharomyces cerevisiae. Key stages in winemaking. Step. Description. Harvesting. Grapes picked at optimal ripeness measured in bricks. Crushing, pressing. Juice extracted to create must. Inoculation. Natural wild yeast or pure commercial yeast strains added. Fermentation. Occurs at 18-30 degrees Celsius for 5 to 21 days. Clarification. Filtration or racking to remove yeast and sediments. Aging. In stainless steel tanks or oak barrels for months, years. Bottling. Final stabilization and packaging. Properly study the flow chart for wine production. Factors influencing wine fermentation. Temperature. Too high equals yeast death. Too low equals slow fermentation. pH. Optimal range 3.0 to 3.5. Nutrient availability. Nitrogen, vitamins, minerals. Sulfur dioxide. Inhibits spoilage organisms. Beer fermentation. Raw materials. Barley malt starch source. Hops antimicrobial, bitterness, aroma. Water. Yeast. Major stages in beer production. Stage. Details. Malting. Barley soaked, germinated, then kiln-dried to activate amylase enzymes. Mashing. Malt plus hot water starch converted to fermentable sugars. Wort. Boiling with hops. Sterilizes wort. Extracts hop resins and oils. Fermentation. Yeast added. Converts sugars to alcohol and COZ. Maturation. Lagering. Flavor development. Removal of harsh compounds. Filtration and packaging. Carbonation. Storage. Distribution. Carefully study the flow chart of beer production. Distilled spirits fermentation. Examples: whiskey, rum, vodka, gin, brandy, tequila. Process overview. Fermentation of sugar or starch-rich substrates such as grains, potatoes, sugarcane, and fruits. Distillation. Alcohol is separated by heating the fermented broth at 78.5 degrees Celsius, the boiling point of ethanol. Aging. Some spirits mature in oak barrels, developing color, aroma, and flavor through chemical oxidation and wood reactions. Blending and bottling for final market product. Distilled spirits typically reach 40 to 50% alcohol content. Microorganisms in vaccine production. Why microorganisms? Microbes are used because they contain antigens capable of stimulating immune response. Can be cultivated at large scale. Can be genetically engineered for safer and more effective vaccine components. Major types of microbe-based vaccines. Vaccine type. Description. Example: live attenuated, weakened organism that replicates with reduced virulence. Measles, mumps, rotavirus. Inactivated, killed. Organism destroyed by heat, chemicals. Polio IPV, hepatitis A toxoid. Detoxified bacterial toxins. Tetanus, diphtheria. Subunit, recombinant. Only antigenic fragments used. Hepatitis B yeast produced, vector-based. Harmless virus delivers antigen gene. Ebola, some COVID-19 vaccines. mRNA vaccines. mRNA instructs cells to produce antigen. Moderna, Pfizer COVID-19 vaccines. Overview of yeasts. Yeasts are unicellular eukaryotic fungi that reproduce mainly by budding. Most industrial fermentations use Saccharomyces cerevisiae because it is generally recognized as safe. Highly efficient at sugar fermentation, genetically stable and easily modified. Cellular structure highlights: nucleus eukaryotic gene control, mitochondria aerobic respiration, vacuoles storage, pH control, plasma membrane rich in sterols. Industrial uses of yeasts, industry. Yeast roll, food and beverage, bread, beer, wine, spirits, biotechnology, enzymes, vitamins, single-cell protein, pharmaceutical, production of recombinant vaccines, for example hepatitis B, biofuel, bioethanol production,